Today, I'd like to tell a brief story in four parts: therapy, pandemic, memory, and prevention. But first, of course, a brief introduction to the science. The immune system. We have two immune systems: an innate immune system and an adaptive immune system. The innate immune system is the first line of defense against pathogens or germs. It releases signaling molecules that trigger an inflammatory response, and it primes the adaptive immune system. I'm going to quote here Columbia University physician and author Siddhartha Mukherjee, who won the Pulitzer Prize for his book Emperor of All Maladies about cancer, you may recall, because his language is colorful. Quote, adaptive immunity involves two principal kinds of cells. B cells make antibodies against pathogens, and T cells hunt for cells infected by a pathogen. B cells can be imagined as sharpshooters that target a virus with well-aimed bullets, that is to say, antibodies, while T cells are gumshoe detectives that go door to door seeking viruses that are hidden inside cells. Both cell types, B cells and T cells, carry the memory of an already encountered pathogen or germ. These so-called memory cells are triggered when a pathogen reappears, and they can swiftly raise forces to fight it. I raise the question, might these memory cells remember today's novel coronavirus infections a century from now in survivors? I suggest the answer is yes, and more on that soon. What are antibodies? Antibodies are the Y-shaped molecules whose tips can attach Velcro-like onto specific viruses or bacteria or parasites. They are biomarkers of infection, and if sustained, can confer long-term immunity. We have billions of B cells that can produce 10 to 20 million distinct types of antibodies. In all, we have the ability to generate 1 quintillion, that's 1 with 18 zeros behind it, antibodies, which gives us the ability to neutralize a vast number of pathogens or germs with protective neutralizing antibodies. And here you see it illustrated the Y-shaped antibodies attaching to the spike protein of the coronavirus, and by attaching, preventing the virus from attaching to the human cell receptor. In this case, the ACE2 receptor, highly expressed in lung epithelial cells, airway, in the airway, but also in other tissues and organs. And this is the entry point for the coronavirus. The virus attaches to the receptor, goes through various conformational changes, and enters the cell, and then releasing its RNA into the cell, and essentially commandeering the cell to do its bidding, and putting the cell's own interests aside. All right, I'm going to suggest 3 never-befores here, and I'm happy to be challenged on this. These are simply propositions. Never before have vaccinated human bodies created so many antibodies in a shorter time in history of vaccination. 600 million doses of influenza virus are made annually. We're going to have billions of novel coronavirus vaccines doses. Never before in the history of medicine has a virus been under greater research scrutiny in such a short time. And never before has the well-being of the world economy hinged so much on a biomolecular binding event. In this case, how tightly the SARS-CoV-2 coronavirus binds to the ACE2 protein receptor in human lung and airway epithelial cells, because binding affinity correlates with transmission. All the new major variants have a mutation called E484K that allows the variants to bind tighter to the ACE2 receptor, therefore enhancing transmission. The novel coronavirus ACE2 receptor binding event has added an estimated $24 trillion to global debt over the past year. This event last May was quite interesting. It brought together biomedical science and finance at the highest level. The Federal Reserve Bank of Minneapolis hosted the panel. The keynote was given by Mr. Jeremy Farrar. Osterholm gave opening remarks. A former FDA commissioner was one of the panelists, as was a former CDC director, and Lawrence Summers, the professor emeritus of economics at Harvard and a former U.S. Treasury Secretary. He said in this hour-long discussion, a vaccine breakthrough or therapy breakthrough is worth it at any imaginable price. In rough numbers, this is costing the American economy $80 billion a week. Again, this is last May. He and his colleague, Harvard economist David Cutler, health economist, suggested in a paper last fall that from March 2020 to October 2021, we could expect to spend $16 trillion. That's first 18 months. Half of it in lost output and the other half in healthcare costs and other associated costs. Bear in mind the floods, wildfires, droughts, hurricanes, polar storms, etc. are measured in the billions of dollars. So an order of magnitude practically smaller. America spent $4.8 trillion in today's dollars fighting World War II. Over the past year, America spent over $5.5 trillion fighting the pandemic. Last month, in a New England Journal of Medicine podcast, Sir Jeremy Farrar, director of the Wellcome Trust, said, when you look at the SARS-CoV-2 virus, it would seem from structural biology mapping studies that the virus still has quite a long way to evolve potentially. The affinity between the virus and its human receptor, the ACE2 receptor, is not optimized as well as it potentially could be. You could increase the affinity of that binding site more than the current mutations have given it, unquote. So now we're going to talk, this is the first part of my four-part story, therapy. Antibodies enter the practice of medicine. This story of Henry Wellcome, I wrote in the early 1990s. I put it up on the web in 1995. I was never able to get it published, but it seems more relevant all the time, especially given the pandemic. Henry Wellcome's first of his many humanitarian deeds was to manufacture diphtheria antitoxin and distribute it at below cost. For the first time, the strangling angel of children could be treated. Let's travel briefly with the long view, beginning with Wellcome's birth in a log cabin in Wisconsin to his boyhood canoeing on the Watonwan River that flows through Garden City, south of Mankato in the 1860s, to working in his father's drugstore and hanging out with his physician uncle, to being his uncle's friend William Worrell Mayo's tutorial student in chemistry in Rochester, to graduating from the Philadelphia School of Pharmacy, the nation's best, in 1874, to the founding with fellow student Silas Burroughs, the Burroughs Wellcome Company in London in 1880, to laying a research foundation for the drug making industry, to building tropical medicine research and care facilities in Africa, to canoeing on the Thames River in London, to amassing vast collections of medical books and antiquities, to sending his medicine kits and branded drugs with the great polar explorers, with Teddy Roosevelt out west, with Charles Lindbergh in his cockpit, and after his death, to the moon with the Apollo 12 astronauts, to leaving all his company's stock to charity upon his death in 1936. The Wellcome Trust now has $42 billion in assets, and it distributes $1 to $2 billion in annual research funding every year, making it the sixth largest founder of health research in the world. And as I've thought this over today, it's as if Wellcome's spirit is out there canoeing on the River Cam in Cambridge, UK. How so? Well, the Cambridge University-affiliated Wellcome Sanger Institute has sequenced more novel coronavirus genomes than any other single research organization in the world. And of course, sequencing the virus is critical for tracking viral variants and identifying any possible, quote, vaccine escape mutants, unquote, that might emerge. Diphtheria antitoxin, developed in the late 19th century, the anti-treatment for diphtheria that Wellcome manufactured and distributed was pioneered in 1890 by the German physiologist Emil von Behring, the savior of children, together with Japanese physician and bacteriologist Shibasaburo Kitasato. Their breakthrough research represents science of pathology, the organized study of the immune system. Von Behring was awarded the very first Nobel Prize in Physiology of Medicine in 1901 for his work. Today, the CDC, Center for Disease Control and Prevention, stockpiles horse plasma-derived diphtheria antitoxin. The CDC's supply is made in Brazil as the U.S. has no domestic manufacturers. And scientists are working an alternative. This therapy has remained largely the same for all these years, and yet we have technologies now that the horses are not harmed, but even so, it's time to move on from horses. I think most of us would agree. Okay, the next part of our story, pandemic. As a side note, last year, 2020, was the deadliest year in recorded U.S. history, according to new figures from the CDC. The 15% increase in the American death rate is the largest since 1918, with the outbreak of the pandemic flu. By the time the influenza pandemic ended in April 1920, it had taken the lives of some 675,000 Americans. In the upper Midwest, the influenza launched its deadly rampage at the Great Lakes Naval Station, just outside Chicago, in September 1918. A month later, the Chicago Tribune, as you see here, reported that the Illinois Influenza Advisory Commission was roused to high enthusiasm by a therapeutic breakthrough. And I'm quoting now, in a word, the treatment consists of injection of a serum extracted from the blood of persons who have recovered from the influenza pneumonia. And again, that's of course convalescent plasma. The Commission said it would try to secure a supply of the precious serum for local cases for use in cases of severe illness by drawing on the graces of the donor volunteers among the jackies, that is to say, the sailors at the Great Lakes Naval Station, and the soldiers at nearby Camp Grant, which is another infection hotspot. And it stresses the serum is absolutely different article from the vaccine, which is to be manufactured under the Commission's direction after the method of Dr. E.C. Rosenau of the Mayo Foundation. That's in this newspaper account. The vaccine is a preventative, and administered to persons who are well. The serum is a cure. They use the term cure here, of course, inaccurately, and is administered to sick persons. By the way, E.C. Rosenau was one of Mayo's great medical minds in the first half of the 20th century. Of course, the blood plasma of the influenza survivors was not at all a cure, it was a treatment. But in 2006, a meta-analysis of clinical studies performed at that time, that is say 1918, 1919, suggests that neutralizing counterattack by the plasma's antibodies may have led to, quote, a clinically important reduction in the risk of death. Now, convalescent plasma is back in the COVID-19 era. It is a type of passive immunity, which is a long history against viral and bacterial diseases. Again, is the standard of care in treating diphtheria. Today, antibodies and transfused convalescent plasma from individuals who have recovered from the infection in patients is used in patients with COVID-19 disease. Last spring, the Mayo Clinic's Michael Joyner headed up a federally funded FDA expanded access program set up to coordinate the collection and distribution of COVID-19 convalescent plasma across the country. More than 100,000 patients signed up for the program. And then in August, you may recall, the FDA granted convalescent plasma emergency use authorization. And last month, it limited the EUA to treating patients soon after diagnosis with high titers, high amounts of convalescent plasma. Dr. Cohn will talk more about this. I hope you'll have some questions along these lines. Hundreds of thousands of COVID-19 patients have been treated, but the jury is still out on whether it really works. How did outbreak occur in Minnesota? It occurred in Faribault County, actually my county. I'm from Blue Earth, you can see down there in the lower left. On September 18th, 1918, when Army musician, Private Raymond Paulson stepped off the train in his hometown of Wells in Faribault County, he wasn't feeling all that great, but he didn't give it much thought. Soon after getting to his parents' house, a telegram came from the Navy. The telegram said Raymond's 22-year-old brother, Walter, had died from a strange pneumonia, and his body would soon be shipped. The day after Walter's funeral, Raymond died, and the day after that, his sister died, as did in time the pastor who conducted Raymond's funeral. The infection spread to the other communities, Blue Earth, Winnebago, largely via the railroad. by the time it was over, 1,000 people had died, more than 1,000 in Faribault County, more than 10,000 in Minnesota. There was an article published in 2007, co-authored by Ruth Linfield, who's the state epidemiologist here. It is astounding to see how you, as much as you'd like to prevent chaos in terms of messaging, in terms of practice and policy, it's very difficult to do. Minneapolis and St. Paul went sort of different directions in many respects with respect to the gathering in public places, how many people in the streetcar, whether you could ride the elevators, the schools opening and closing, and on and on. I think they were simply conducting an experiment and looking at each other. The case fatality rate in Minneapolis was 5. The case fatality rate in St. Paul was 15. So when you have a pandemic, you're going to have, even with the best planning, you're going to have some chaos. The article starts off with a famous quote from George Santayana: "Those who cannot remember the past are condemned to repeat it." And then after reading the article, I was reminded of the famous quote from Ecclesiastes: "There is nothing new under the sun." The third part of the my 4-part story is memory. How long does memory last? In 2004, Eric Altschuler, a medical resident, was watching television, a television show one night while he was on call. It was a slow night, as he put it. The show was a short-lived TV series called Medical Investigation and featured fictional scientists who tracked down causes of mysterious diseases. In this particular episode, Mutation, a team of NIH scientists flies to a small Virginia town to figure out why people are dying from a deadly strain of flu. Through contact tracing, we've heard that term recently, of course, and asking questions, they find that the deadly flu spares an elderly butler who had survived the 1918 flu pandemic as a toddler. Anyway, they fly the butler to the NIH and draw his blood, prepare convalescent plasma with his antibodies against the H1N1 influenza virus from his blood, return to the scene, and give the infected people the butler's convalescent plasma just in time to save their lives. However, there's not enough convalescent plasma to go around, so rationing is implemented. Of course, you remember the term rationing in the context of ventilators last spring. Anyway, Altschuler wondered, might immune system memory still be present in the elderly survivors of the 1918 influenza pandemic? And so he approached the NIH. Marcel Proust, Remembrance of Things Past. There's no artist who's more associated with memory than Marcel Proust. He published his 1.5 million word, 7-volume masterpiece, Remembrance of Things Past, or more recently translated as In Search of Lost Time, from 1913 to 1922. The English writer Graham Greene called Proust the greatest novelist of the 20th century. And he's remembered, if those who have not made their way through the 7 volumes, he's remembered mostly for 2 things. The Madeleine cakes. He takes a bite of a Madeleine cake in Swan's Way, the 1st volume, and suddenly the memory returns from the vast structure of recollection. So the memory, the Madeleine cake brings forth a flood of childhood memories that he writes about in amazing detail, and that's called involuntary memory. The second thing that he's remembered for is his cork-lined bedroom. As an adult, he spent most of his adulthood in bed, in the bedroom, cork-lined bedroom, because he was an asthmatic. He was a hypersensitive to any environmental assault, and he would sleep during the day because there was a lot of commotion outside. At night, he'd sit up in bed, draw his legs up, put a platform on his legs, and write. And he'd write all night. He was as had been described, the original master of social distancing. Perhaps not so surprising, because his father was the French epidemiologist and hygienist Adrien Proust, who himself pioneered concepts of quarantine, cordon sanitaire, containment, and social distancing, described as the man who wanted to confine everyone. Adrien Proust was dubbed the geographer of epidemics, based on his travels tracking epidemics abroad. In December 1918, the flu was there in France, and Proust wrote to the editor of Nouvelle Revue Française, "Dear Madam, I would be pleased to know if you and your son and your loved ones, as well as everyone in the review, escaped the flu." Okay, the question is, where does the memory of 1918 influenza pandemic reside in today's survivors? Well, in the brains of some who can recall, those few who are alive and can recall, and in the memory B cells, and perhaps all people who were exposed to it who are still alive. And so in 2008, a study led by Vanderbilt University investigators, including Altschuler, studied 32 pandemic survivors ages 91 to 101 for biological memory of the influenza pandemic to which they had been exposed. And there were controls. They also studied people who were born after the pandemic. They wanted to determine if the survivors still had antibodies to the virus, and to see if the B cells that produce the antibody could be cultured and induced to make antibodies to the key 1918 virus protein. They concluded, these studies demonstrate that survivors of the 1918 influenza pandemic possess highly functional virus neutralizing antibodies to this uniquely virulent virus, and that humans can sustain circulating memory B cells to viruses for many decades after exposure, well into the 10th decade of life. So when I would raise the issue, a century from now, those still alive who've been exposed to coronavirus, will they have the memory? Well, their B cells will, probably, based on this study. And it's not just B cells that have memory, T cells have memory as well. And in fact, Siddhartha Mukherjee, I mentioned earlier, just published a piece in the New Yorker magazine. Why does the pandemic seem to be hitting some countries hard? Subtitle, while the virus has ravaged rich nations like the US, reported death rates in poorer ones remain relatively low. What probing this epidemiological mystery can tell us? And he suggests, based on the comments of some scientists, that the T cells in people in these countries may have been exposed to previously been exposed to other types of coronavirus that produce only mild symptoms. There are 3 or 4 of them, and they may be more prevalent in some of these countries. And they therefore, with exposure to these other coronaviruses, they have built up some cross-reactive immunity, and maybe that's the reason. Again, with the memory T cells coming into play. Okay, and this is the final section of my talk about, in this case, about prevention, the pandemic influenza vaccine conundrum. In the Twin Cities in 1919, there were 2 different vaccines, neither of them effective, because neither was informed by influenza virus. They were informed by bacteriology. One was made by the University of Minnesota, a University of Minnesota bacteriologist, was purported to prevent pneumonia. The Mayo Clinic made another vaccine that was intended to prevent both pneumonia and influenza. These vaccines were premised on Pfeiffer's bacillus, a bacteria first described in 1892 by the German physician and bacteriologist Richard Pfeiffer during an influenza pandemic. He saw these rod-shaped bacteria and associated with these infected patients, and made the assumption or the hypothesis that these bacteria were in fact causative. So Pfeiffer's bacillus was argued by some to be the cause of influenza until 1933, when the viral nature of influenza was firmly established. And infection still as bacteria influenza, now known as influenzae. The bacterium is responsible for a wide range of localized and invasive infections. Anyway, the vaccines, bacterial-based vaccines were widely distributed in the Twin Cities. Military personnel as well as civilians were inoculated beginning as early as October 1918. Both city health departments purchased vaccine and distributed to physicians at no charge to encourage widespread use. And this is again from the 2007 article, of which Ruth Linfield, state epidemiologist, was one of the co-authors. In Minneapolis, people desiring the vaccine thronged to the offices of doctors hoping to be vaccinated. And in St. Paul, it was reported that, quote, thousands of persons have been inoculated, unquote. So that's the conundrum dimension here is that these were, this is a bacterial, antibacterial based vaccine, purported vaccine. But in fact, you may have heard that for those who died in the 1918 influenza pandemic often died of bacterial pneumonia following viral infection. And the question is, might these vaccines based on bacteria, might they have been useful in treating those who develop bacterial infection following viral assault? And we don't know the answer, but there is the suggestion in the scientific community. In fact, this might have been a good thing to do. All right, we're in the midst of a revolution here, and you undoubtedly know about the revolution in mRNA vaccine technology. It is astounding. I've been working in the medical school since 1983, and I've seen quite a lot of innovation. I've never seen anything quite like this, though. And I'll give you a time frame here. January 10th, the SARS-CoV-2 coronavirus sequence is reported to the World Health Organization by a Chinese scientist. 30,000 base pairs and 26 genes. By the way, we have 3.2 billion base pairs, we humans, and 20,000 genes. Okay, that's January 10th. 5 days later, the NIH designs mRNA vaccine in collaboration with Moderna. March 16th, 2 months later, Moderna human trials begin. November 16th, 8 months later, Moderna reports phase 3 human trial results, 8 months. And then the next month, December 18th, Moderna vaccine receives FDA emergency use authorization. And of course, the Pfizer BioNTech vaccine, mRNA vaccine, was actually approved earlier, a bit earlier. But it's essentially the same story. The Wall Street Journal wrote about this new platform, and I'm quoting now: researchers in gene-based technology produced a class of vaccines they believe can protect against all manner of outbreaks in the years to come. So we'll see about that. Okay, I'm wrapping up now. Ecology and health. Again, Sir Jeremy Farrar, just last month. The pandemic is the end result of drivers which are going to remain, in my view, through the 21st century. They are changing ecology, changing land use, changing interactions between humans and animals, and then humans living in huge cities, densely populated, highly connected with both the country and globally. My virtual background, you notice that this is my virtual background, is Cedar Bog Lake at the U of M Cedar Creek Ecosystem Science Reserve in East Bethel, where graduate student Raymond Lindeman did his pioneering ecosystems ecology work, field work in the 1930s. And I recently wrote a book chapter about Lindeman entitled Raymond Lindeman, Minnesota Bog Lake, and the Birth of Ecosystems Ecology. The term ecosystem had just been coined when Lindeman began his field work on Cedar Bog Lake, but Lindeman gave the term a firm scientific foundation, published a very famous paper in 1942. And as the Wellcome Trust Sir Jeremy Farrar frames it here, ecosystems have everything to do with pandemics. In our 2014 book, The Biologist's Imagination, Innovation in the Biosciences, Leo Furcht and I published that. We use this epigraph: the ability of human societies to modify and transform systems will increase more in this century than it has in 100 centuries since the dawn of agriculture. That's from the journal Nature. And I pose the question here to you all: What about the ability of biological systems and ecosystems, of which pandemic viruses are part, to transform us? Pandemics change societies. The current pandemic will change us in ways we cannot now imagine. The long view from the river will express how we see ourselves after we emerge, how we illuminate the journey through and beyond the pandemic in imaginative and creative ways. As Marcel Proust once wrote, time is preserved in memory, and memory is preserved in art. And some words of wisdom here from Chief Seattle. I'll stop there and ask for Claudia to come on board, and please do send us some questions.
I have a question.
Yes.
Doesn't this call into prominence the importance of history and the remembrance of, like you said, the remembrance of things past, knowing what worked in the past and not having to call up, not having to redo everything all over again every time.
Of course, that's it's going to be interesting. This will change us in some ways. Whether it will change us in ways that prepare us for another, God forbid, experience like this, it's really hard to say. The notion that these are 100-year pandemics, I think we can put aside. Just based on the forces at work, bringing us into contact with zoonotic diseases, what they call zoonotic diseases, and the proximity we have with animals who may be reservoirs, such as the horseshoe bat in China. The horseshoe bat, according to some experts, has an untold number of coronaviruses that circulate in its system, where, as one put it, we're in B in the alphabet when it comes to coronavirus and horseshoe bats. Now, most of them are going to be mild, probably, and not serious to humans, but you never know. And of course, we don't know now with the current coronavirus. We don't know, although we're given some reassurance. I follow this, that we won't find there won't be a mutation as deadly as, let's say, that would produce levels of death, severe sickness and death, that the 1918 influenza pandemic produced. And of course, if the virus develops in ways that allow for tighter molecular binding, that will enhance its transmission. And over time, of course, greater transmission means more sickness and death in the long haul. But of course, the vaccines are coming on, and the idea is that right now the vaccines. You may have heard about this. I'm getting off Rich's point a bit. The vaccines look to be effective against the mutants that are out there, and the antibody production may be slightly diminished. But don't forget, the T cell memory is critical for protection as well, not just the antibodies. And there hasn't been a lot published or discussed about T cell protection following vaccination or natural infection.
I have a question.
Yes.
Yes. This is regarding T cell memory.
Yes.
Okay. So I'm a retired pediatrician, and I was exposed to coronaviruses every day for over 25 years. And I've been wondering, from what your comment about other nations where they may have had more exposures, if any studies have been done, because it's the only adult population that's been exposed to coronaviruses on a daily basis for many years. Any studies have been done on how many pediatricians have gotten coronavirus or their T cell?
That's a fascinating, yeah. It's a fascinating question. And it, my guess is, but thinking back to this, the TV program that Eric Altschuler was watching, I would think that this is going to have to be pursued. I don't know the studies are underway, but Dr. Cohn may know something about that. Are you there, Claudia?
Yeah,
okay,
it's a great question. I've not read of any studies that specifically target the pediatrician population or other populations that would be at a higher risk of exposure to related coronaviruses. I know that there are basic science studies and of how exposure to certain viruses in the past can actually promote a negative response in people. The classic one is if you've been exposed to dengue virus and then you are exposed to Zika, that you have a much worse response to Zika than people who have not been exposed to dengue. And there are people who think that this idea of antibody-dependent enhancement will occur due to past exposures to coronavirus, but it's not been shown on a population basis or in a lab yet. So no, I think it's a really good question, but I don't know the answer.
Okay.
We have another question from Bob that says that you seem to say that the world would be flooded with billions of doses of vaccine. Is that correct?
Well, I can say that based on what I've seen, that Pfizer and Moderna, J&J, AstraZeneca, Novavax coming on, we are talking about billions of doses in a short time. After all, the first doses were outside of clinical trials, once approved, were just late last year. And so if you go from late last year to late this year, I am sure that we are going to be talking about billions of doses manufactured. How many administered, we'll have to wait and see. The annual manufacturing of influenza is 600 million doses, of which a fraction of use, about 50%, are actually administered. But 600 million doses versus billions for coronavirus. I think that's a reasonable estimate.
Okay, thank you. How soon do you expect booster vaccines to be required?
I'm sorry, beg pardon?
How soon do you expect booster vaccines to be required?
The CEO of Pfizer says a booster can be produced in about 3 months. That was a comment he made a short while ago. They are working on that right now, as I understand it. Presumably, maybe it was a month ago that he said that. I'm guessing that they will have a booster capability in a fairly short time, but I don't actually know. And the thought is that come fall, some of the authorities think that boosters will be necessary, or at least handy, and there should be no problem. The vaccine manufacturer is ramping up very quickly, and even though we may not have everything we want in March, they're talking April and May as being abundant, even potentially oversupply of vaccine. Now we're talking about the advanced. We're talking about advanced economies, the richer nations. The biggest challenge, of course, is to make the vaccines available to developing nations, because we really are in something like this all in it together. I mean, if you've got widespread transmission somewhere, it's going to, dangerous mutants are going to find their way out and in our presence. So.
Do you think it will be necessary to have annual vaccines?
That's something I can't answer, but I have heard and I have use the term endemics, an endemic like experience. And others, people who are in a position to know, have said the same thing, that this is a possibility. It would be an endemic situation, a controllable, manageable virus. We're not there. It is not the flu, but the possibility of it becoming manageable with vaccinations and boosters is, I think, potentially on the horizon. Yes.
Do you see an increased interest in producing vaccines and drugs in the U.S. rather than abroad as a national security issue?
Well, that's a very good question, and it's the national security world struggles with biology in some respects. It doesn't seem quite like it wants to. I mean, don't get me wrong. There are agencies within the government and the military that are looking at biology very closly. In our book, The Stem Cell Dilemma, published in 2008 and second edition 2011, we have a chapter on DARPA, the Defense Advanced Research Projects Agency, which brought us the GPS, brought us the internet, brought us predator drones, and so on, getting into biology, and in fact developing a tabletop rapid vaccine assay system based on human cells, so that you could test a vaccine on in the machine rather than in human trials, in order to do it in a rapid way in case there was a biological weapon developed. So the answer is that national security has to take this more seriously, and the authorities will say that you can't just think that you protect people in advanced countries and that you're going to be okay. Pandemic is a collective. It is a collective all the way through, and the reason that New Zealand, they're talking about that now. There's an article in the paper. The reason New Zealand has such a positive experience with this awful pandemic compared to us is maybe in part that they've adopted, the New Zealanders, the Maori ethos of the collective, especially during times of stress. That the collective is the key, not the individual. We have a somewhat different way of looking at it, and we're paying the price.
A comment from Rich McKay is not just the vaccine supply chain, but all aspects of the U.S. supply chain has been exposed during the epidemic. Note the difficulty of procuring PPE and right. The national security flaw of just-in-time supply chain has been exposed.
Right. And Mike Osterholm of the U of M has been talking about this certainly during the whole period of the pandemic, especially early on. And his 2017 book addresses this. How can we allow the supply chain to move beyond our control and our ability to influence? And so, any number of key pieces of equipment or the foundation agents for drugs, etc., are not made in the U.S. They are made overseas. And this is all going to have to change. And it's going to be quite a dramatic, I think, at least in the short term. Who knows about 20 years from now? But in the short term, there's going to be major change that deals not only with supply chain issues, but with the whole vaccine world. Up until this experience, nobody wanted to go there because the vaccine is sort of one and done, and nobody could make any money. Well, this is going to be a massive governmental effort, collaborative effort with private industry and funded by the taxpayer, so that we don't run into this again, at least in the near future.
A question: some people who have gotten mild cases of COVID have gone on to develop long haul symptoms.
Yes.
A small number of vaccinated persons can be expected to develop mild cases of COVID from subsequent exposure.
Yes.
Is there any data yet to indicate whether vaccinated persons who later contract COVID are or are not at risk to develop long haul symptoms?
I don't know the specific answer to that, but I can say that I just read a story about how vaccinated long haul patients have improved symptoms. Okay, they're a long haul is can have a whole array of health problems associated with it, many different organs involved, and I just did read a story that those who've been vaccinated, those long haulers who've been vaccinated, are having significantly reduced symptoms compared to those who are not. So I could say that it would be worth looking into. I can't answer the question more specifically than that. But the long hauler, we've only had this for a year now. So we know the long hauler experience for 1 year, but we don't know it for 10 years. And when I mentioned Summers and Cutler's article on the $16 trillion cost over 18 months, they're figuring chronic disease. They're taking into account the cost of chronic disease, and it could be much bigger than maybe even they are estimating.
Okay. After 2 Moderna or Pfizer vaccines, how likely is it that one develop COVID-19?
I don't know. After vaccination?
Yes.
I don't know the answer to that. I can tell you that that all the vaccines seem to be almost 100% or close in not preventing infection, but in preventing serious disease, hospitalization, and death. So these vaccines may not prevent infection in everyone, but their ability to keep you out of the hospital is just remarkable. And that's, of course, what we are really interested in.
Paul Scanlon commented, he said, thanks, Bill. After Moderna or Pfizer, risk of mild COVID-19 is 5%. Risk of severe COVID-19 is near 0. Yeah,
you are. That's it. Go with Dr. Scanlon there. But I knew it was very high. And of course, that's, the idea here is to protect everyone against severe disease, hospitalization, and death. If we have to put up with a little flu, of course, you may have to do that even with the vaccination, and some side effects. That we can survive some time of that kind of discomfort. It's the complete breakdown of the respiratory system, and et cetera, and the long hauler complexities we want to avoid. So there is no experience in the history of vaccination like the one we've had to have these safe and effective vaccines produced in such a short time. And the mRNA platform is going to be implemented for not only for dealing with future viral infections, but for the whole range of what used to be pharmaceutical interventions. It'll be gene-based therapies for many things.
Paul Scanlon also commented that today's Trib has an article about long hauler syndrome.
Yeah, good. It's just a judgment I have that the costs associated with that, certainly the costs of patients and their families, but the monetary costs are we have not yet begun to appreciate how significant that is, and in terms of workforce participation, for example. So that's an economic issue right there. So you want to get these vaccines out there and get them. I've had my first. I got my second at the end of the month, and I'm more comfortable now. I've got I'm 2 weeks out. I've got some antibodies going here, and in theory, at least, I probably won't end up in a ventilator if something does bad does happen. So I hope anyway. So.
Does anyone have any other questions, or Dr. Cohn, do you have anything else that you would like to add?
My angle on COVID has largely been from about convalescent plasma, and I've been following with great interest all the confusing studies that have come out. The jury is still out, as Bill already noted, about whether convalescent plasma is helpful, although it is my interpretation of the data that if you have developed symptoms of COVID and managed to get convalescent plasma within the first 3 to 5 days, really the first 3 days after symptoms develop, that you will receive benefit from the convalescent plasma. That is, the data are strong for that. Otherwise, if you're in the hospital and quite sick and receive it, probably no benefit.
Yeah.
So earlier is better. But that's my only comment. Thank you for this wonderful talk, Bill.
Thank you. Thanks for joining us, Claudia. Bye-bye.
One more comment from Rich McKay. In 1918 epidemic, was there opposition to masks and preventative
Yes.
and preventive measures? Yes.
There was, and that includes in the Twin Cities, Minneapolis, St. Paul. So the mask, the great mask debate did occur back then too, and there were mandates, and there were depending where you were. The masks were gauze masks those days, not N95s. They were gauze, and I don't know, but I'm quite sure that gauze masks provide marginal protection, not great. But if you see the photos of the 1918 pandemic, people wearing masks, but the masks are pretty. They're pretty poor quality for protecting against H1N1 influenza transmission. The N95s, of course, are very good, not perfect, but very good. And then not everybody has those now. We manage with the masks we have. So.
Okay. Any other questions from anyone? Okay, well, thank you so much, Bill, and thank you so much, Dr. Cohn, and everybody for joining us. This was fascinating and informative, and I think we're all a little bit in better place as far as knowing about COVID and about the history. And it was a pleasure to hear your comments and to learn all about this. We have several thank yous on the chat. I don't know if you can take a look, Bill.
Well, thank you.
But other than that, and the slides were wonderful. So thank you everybody for participating, and it was fascinating. Thank you.
Thanks so much.